Welcome to Biot Anime. Molecular chaperonins. Molecular chaperones are diverse families of multi-domain proteins that have evolved to assist nascent proteins to reach their native fold, protect subunits from heat shock during the assembly of complexes, prevent protein aggregation or mediate targeted unfolding and disassembly. Different molecular chaperonins in the system are 1. HSP-60, 2. HSP-70, 3. HSP-90, 4. HSP-100, HSP-60. HSP-60 family of chaperones or chaperonins is key component of cellular chaperone machinery. They are the most conserved and ubiquitous class of a molecular chaperone present in the plastids, mitochondria, and cytoplasm of all eukaryotes and eubacteria. Chaperonins form oligomeric, high molecular weight complexes of approximately 800 kD. It forms a large cage-like structure formed by two heptameric rings, each enclosing a central cavity. The heptamer is made of similar subunits. The chaperonins are classified into two groups that are structurally similar and quite diverse in sequence. The group I chaperonins are found in prokaryotes and endosymbiotic organelles such as mitochondria and chloroplasts. Group 2 chaperonins exist in archaea and eukaryotic cytosol. Group 1 chaperonins. The group I chaperonins are found in prokaryotes and endosymbiotic organelles such as mitochondria and chloroplasts. Group 1 proteins include GROEL in bacterial cytosol, HS60 in mitochondria, and rubisco-binding protein rubiscoBP in chloroplasts. Group 1 chaperonins employ a detachable lid structure called GROIS HSP-10 that binds in an ATP-dependent fashion. A good example of group I chaperonins is the paradigmatic Escherichia coli GROEL-GROIS system. It consists of 14 subunits arranged in a double heptameric ring complex and requires a ring-shaped cofactor GROIS for its function. Each heptamer encloses a central cavity that forms the folding chamber for the polypeptide substrate. The constituent subunits of the heptamer contain three domains. The equatorial domain harboring the ATP binding site, a substrate binding apical domain, and a middle hinge domain that enables communication between equatorial and apical domain. The apical domain forms the opening the GROEL cylinder and exposes a number of hydrophobic residues towards the ring cavity for substrate binding. The ATP binding to GROEL occurs through allosteric mechanisms. Prior to ATP binding, both the rings are in the tense state. Negative cooperativity allows the nucleotide to bind to cis ring, converting it to relaxed state and lowering the nucleotide affinity of the trans ring. ATP binding causes substrate binding and binding of GROIS. This allows establishment of intramolecular interactions in a protected aggregation-free environment. Hydrolysis of ATP in the cis ring followed by nucleotide binding in the trans ring cause dissociation of GROIS and substrate release. Group 2 chaperonins. Group 2 chaperonins constitute thermosome TF55 in archaea and tricked CCT in the eukaryotic cytosol. Group 2 chaperonins employ a built-in protrusion structure which may be either marginate or open upward in a polypeptide accepting state and that then closes when the ring binds ATP to produce the folding active encapsulated state. TRIC is expressed constitutively and is required for folding of essential proteins. The emerging polypeptide chain is transferred from the ribosome to TRIC via either the chaperone GIMC or the HS70 chaperone machinery via prefoldin. The apical domain of TRIC has a long extension, which forms alpha helical protrusion. This extension is highly conserved in the TCP1 family and contains hydrophobic residues that act as an substrate binding sites. The extensions arch over the central channel to form substrate binding surface and act as a lid. Thank you for watching the video. For more videos, please subscribe to this channel and press bell icon. See you again in the next video.